about health. Okay, that's the next one. Okay, so these are the drugs, repurposing the drugs which already tested in the influenza and cancer and all those. It has, different effect. It's not, some drugs work, some drugs does not work. Some has works for some person, some does not work for other person. So, and you can see those are the targets, where the targets RDRP means RNA dependent RNA polymers, which is the enzyme where the drug is actually affecting. Because for RNA virus, the repurposing is very easy because all are RNA and all have the RNA dependent RNA polymers. So if you one drug like influenza, they have RNA, so why not you try for the other viruses, right? So that's why they tried all those, and it works. Sometimes it does not work. Mostly, yeah, yeah. Okay, so the prevention is you can see like basic thing. It's a basic general knowledge here. Clothing and proper PPE, protective uh, equipment. Okay, personal protective equipments. That's called PPE. So you are going to the field. What is it? Not a Bermuda or not a like short shirt because it's so hot. No, cover your body so that you don't get exposed to tick bites. Same thing when you are abatters. Abatters means those butchers who are cutting the meats and all those. So you protect your body so that if infected animals or something, blood is contaminated, it does not stick. You saw in a Kolkata, saw like goat meat shop, that chicken, right? They're cutting with the lungi, and you can see the foods, all bloods already touched there. Do you think it's a good idea to do this? No, that's not, because you're contaminating yourself. The chicken or goat has so many other bacteria. We talk about like hantavirus, that it is there in the sheep, it's in the cattle, camel. So that way we get transmission also. So basically it's covering your body. You can control the ticks, use the vaccine, education, which is, I think it's more important in quarantine. If you see some, they are stick infested, animals, they are just keep it separate and all those. There are several vaccines approach has been, name it, they have tried recombinant, transgenic plants, okay? The animals, there's another lessons, one my friend was working transgenic plants. So the plants also, you can put those viral proteins there, and the plant leaves will express those proteins. And when the animals are consumed, or even humans, they consume, they get that protein, and you can generate those. So antibody against those. So, VLPs, viral-like particles, DNA, so mRNA vaccines. So they tried all those in different animal model. The protective efficacy demonstrated by vaccine only found out the nucleoprotein and glycoprotein, which we have shown in the structure. The glycoprotein and nucleoprotein are more immunogenic. The correlates of protection. What is correlate means what is actually giving protection, whether it is T cells or B cells or antibody. It is not known yet, because and the neutralizing antibodies are dispensable. So it's come very late. You saw that, right? The antibody comes very late when the person is already recovering. So may not be the neutralizing antibody is doing something there. So, but still, studies is going on. The role of cellular immunity, like T cells, is also unknown. So T cell, B cells, not doing too much that early recovery. Who is doing it? It does not know. So it's still studies going on. And as the underlined, there no commercialized vaccine. So again, this is very important, and it is prevalent in India. You can see, check the reports, Times of India or any newspaper, the Hindu, its outbreak happen all the time. It's a tick infested, that's major important, and there is no vaccine, and it needs BSL-4 like environment to study. Okay. Um, I think that is the last. Oh, so you can see that cases. India, Russia, all those Africa, it's not there in America or anywhere, but it's mostly Asian country, African countries is very prevalent because the ticks are there. That is the tick zone. Those ticks are everywhere. This is the major ticks. There are 30 ticks, but major is hyaloma. Hyaloma is very popular in India. So those are the ticks which actually do the transmission. A lot of livestock can be infected because the livestock are the reservoirs. Outbreaks are focal, human infected from tick bites with or removal of the ticks or contact with blood or tissue of infected livestock. That's why the abattoirs, right? Cattle already, the cutting mates, they are maybe infected with all this premium Congo. So that's why you have to take care of the shepherds, ranchers, abattoir workers. The nosocomial outbreaks also occurs. So this is the life cycle. The small mammals and birds. So this is the major, the ticks. And the ticks, when they gives birth, it's also transmitted to their babies. So it cycle all the time. Is the baby is also infected. The baby when mature and give birth, they are also. So its cycle is going on. But also they infect the small mammals and birds. Also all those livestock like sheep, goat, cattle, those, and humans get those either tick bites or from those animals when they are handling. Case fatality rate is 5 to 30%, usually 5 to 14 days after onset, so that the maximum 30% death happens. And it's endemic areas reported lower case fatality. So endemic areas has a lower case fatality, but the mostly is 5 to 30% death happens. And so improve laboratory capacity, geographical expansion of virus through movement of ticks, humans, because humans are moving from one place to another. Ticks also all over the places, livestock, you have a Bangladesh, you have a goat from a cattle from Bangladesh and brought it here. So I want to, but the cattle already infected and he just transported that virus also with the cattle to your place. And that's it. Can you please put the next folder? He knows. So next we'll be talking about Kyasanur forest disease. How long? Maybe this one and then stop, or I don't know.
Week of time, I think,
Oh, no. So then you can start the Ebola also. Okay.
Ebola we want to clear.
Okay, so the Kyasanur forest disease. This is more prevalent in India. And I selected, so again, remember, this is a flavivirus. So from these pictures, you can see this virus is always present in the monkeys. India is famous for monkey, Hanumanji, right? But the monkeys, oh no, Hanumanji, that is the separate religious aspect. We always believe in religions, and I also am very religious. But the primates, which always feed them, oh, they are so lovely. But you are getting the virus from them too. so, so I from those. This is another reservoir. Monkeys are the major reservoir of this virus, and this is the again tick, flavivirus tick. They are the transmitting those disease. This is the map of India, Tamil Nadu, Maharashtra, the Karnataka, all those Gujarat, Bengal, Kerala. They are all KFD. They are positive and likely to spread the other state like Madhya Pradesh. I think, yeah. So these are those Hyderabad. So they're the places where it's spreading also. This is the so far the reported cases what they found. So 1957 in Shimoga they found like 9 cases, and you can see the list till 2020 it increased drastically 448 in Maharashtra. So you can think about it on the in the map we have seen earlier. In Karnataka, a major hit, and then Kerala, Maharashtra, and those they have not listed here. It's few cases, but majority it's the western belt. Okay, so there. So every virus, it's the same principle. The virus goes in, do the applications, uh, and all those. It's more important. The post-transcription modification and virus assembly. So the virus is very intellectual. When post-translational modification, I don't know how many people take, you are in molecular biology, right? You should know molecular biology or read something. We cannot talk now, all those, it will take time. So how the post-translational modification happens, what is the translational or translational modification? So, but the point here that virus can modify the hijack. Cell always do their post-translational modification. Those can hijack that system and try so that they can change those post-translational modifications, and then they make different mutants and all those, and then release those. And these are the things like if we're thinking where we can use the potential drug targets. So when the structural proteins are generating is binding, can you stop that pathway? So people, that is the normal generic ideas all the time for all viruses. Like HIV, people are blocking viral binding receptors. People are blocking protein production. People are blocking reverse transcriptase production. So that RNA dependent RNA polymers. So targeting those pathways just to stop the virus. Here it is how the virus transmission happening. That's the tick. Haemophilus is the major reservoirs for this. And as I said, as they lay eggs, they transmit the virus to the eggs too, and then they become larvae, nymph, and then it also it circulate between. So one adult that tick gives multiple birth, so everybody has that virus. Then everybody giving multiple birth, so it's like they're multiplying and they're circulating within this, and they, stay there. And then when they infect, maybe in a mouse or primates, monkeys, they transmit them. But monkeys also get susceptible. Monkeys dies too. Uh, how many people looked over, right? Monkeys are dying. Who cares, right? Whether they are due to that disease or not. But it is good to study. Like I saw my locality, some monkeys die, like 10 of them. Somebody should study like what is the cause of the disease, why they died. Then we can be sure, oh, all of them found this virus. That means there's some way the transmission happened. Somebody got it somehow. So those things are very important to know. And then again, as I said, feeding those monkeys, and so you're getting those infections. Even the farmers are going to collect the woods and all those, they get the tick bites and get the infections. So the clinical of patient-formed carcinoid disease. So there are a lot of reports as well. You can see and all those the same like meningitis, seizures, then headache, diarrhea, and all those things. But majority, all the reports they have a fever. I mean, it's a viral disease. You should have a fever because always fever is induced by pro-inflammatory cytokines, right? That is always there. Myalgia is majority they have reported, and also bleeding is kind of in between. Diarrhea is present all the time. Headache is there to some extent, but others symptoms it's very low range. Mortality sometimes you can see very high mortality to there is no mortality. So it varies. Okay, some reported okay there is high mortality. Some say oh there is 100% mortality. That here the two-person got infected and all two died. But you see 9 of them, only 22% mortality happened. So that's depends like how much tick infestation happened, how much they got the virus. So those things matters too. Virus goes circulation and it sacrifice does. to induce more adaptive immune response. The T cell and B cell functions comes and ability hemorrhage also. And again, the DIC. So we discussed a lot yesterday, today. So it causes hemorrhage. There's platelets go down. A lot of test has been used. So PCR, we know cytopenia. So those like measuring like CBC, blood count, complete blood count. You're doing all those, and to find out what the virus is. PCR is more important, and IgM ELISA because it's more specific. Like by PCR, what primary you're using, and you detect those viruses. Okay. And vaccine. To give another vaccine, so you never expect that it is give a lifelong immunity forever.
So they paid
We don't know.
the people will die.
Yeah,
Probably they also will have some comorbidities because of that.
yeah. So there are a lot of
It's not fair.
yeah. Mortality is there, but those things we don't know. Like how protection, longer your protection will give, or if you have a secondary infection, maybe you are protected. You don't worry about. Right. So they say the vaccine should be given immediately after exposure, but you also don't know when I got the exposure. So only people think about, oh, I have a fever, then say, oh, I may be exposed to this. So that's the kind of, it's complicated.
Once in every year it will be reported, especially from human. Every year it will be reported. Luckily,
So human to human, it's not reported. Like if some family members, others, they don't report it. It's transmit from human to human. It's mostly from either tick bite, or you're feeding a infected monkey, and you get from them, or or any animals like abattoirs, like it's already cattle infected or sheep infected, get from tick bite, and the blood contaminations. You got the blood contamination because blood has a high level of the viruses. That's where you get, but not as a direct human to human transmission like influenza and other does. So but this is BSL4, yeah. But we don't have India does not have use the BSL4 to study this. It is under, if you see under the CDC, go to the CDC website, cdc.gov. You can find the whole list. What are the BSL4 agents? What WHO and CDC has recommended. So the Kaisener forest is a BSL4 agent. You need to have a BSL4 person. That's why it has not been studied. Sporadic case from the clinical samples, right? Because nobody has studied those. If you want to study, you need a lab. So that's kind of all kind of speculations. Okay, that's why I put the arrows. oh, T cell, B cell generate, okay. Okay, that may be happening. But actual real data, you need a lab to do this, and I don't think done yet in India. So
Why this has been categorized as BSL? Because it's not.
because that is not effective. It causes meningitis. It causes hemorrhage. When it is very related to hemorrhage, it automatically goes to BSL-4 agent because it's causing hemorrhage, and people want to understand why the hemorrhage is happening. So any disease, if you think as a general, like hemorrhagic disease, it automatically
It
go.
doesn't happen in many of the infection or many of the patients that it doesn't go to the brain or.
Well, that is my question too, because how well it has been studied, right? And how, like, a patient came and almost going to die, or maybe what stage you are looking at, how long you are following up those patients. Those are important because it's like a snapshot, right? I just did one sample and those, but there is not too much clear data.
So my question is that like whether infection naturally or ultimately leads to the that means like a brain infection.
It goes to brain.
All the cases.
Exactly. It goes to brain, it goes to hemorrhage. So, it crosses the blood-brain barrier because it causes hemorrhage. When it's hemorrhage, it's more chance to go to the brain because it can cross the blood-brain barrier. Yeah, that's kind of principle there. Hemorrhagic disease go to the brain easily. And when it is there, that's why it causes brain problem. Yes, and the transmission is very low because it's mostly ticks or the tick infected some animals. You get transmission happening. So that's why people don't take it seriously because it's not like flu, so much easily transmissible, measles easily transmissible. So it is, and not too much death. Oh, two died, okay. They may die anyway, right? So they died. So that kind of, but I think this is another disease which, either, control the ticks, and it's very difficult to control ticks, right? I think more education needs to be done, and there is not too much data there. What is happening, doing, how the what are protection? Those question has not been answered. So, and for those you need some kind of animal model, some kind of laboratory where you can do, and that needs BSL-4 laboratory, and those great. Yeah. Okay, I don't know. Next. You said 1:30 or
150.
1:50. Okay. So we'll start Ebola. So it is a big presentations. I recently gave another University of Alabama. So there is a YouTube. So we'll also here. So so there are so far like day 1, day 2, today day 3. I presented few of my data, right? Whatever research I did, and because based on that's why I asked Dr. Reddy like what's the audience is, and as you are undergraduate, I don't want to bombard whatever research and say, oh my, it's so boring, he's talking, I don't understand. So that's why I'm giving more like a generic view, at least give a basic idea what is the virus is, get some idea how the virus replicates, what is the incubation period, how the transmission happens, what are the precaution measures we can take. That's kind of very generic, and I think as a UG level, I think those are important. When you go to, that's why if the audience is here all PhDs, I have to totally give the different weight of presentations because I know all this, what's the new here? So this is the presentation I gave for PhD level, and also faculty level. But bear with me, if you have any questions, we can answer those. Some of those also I presented with my own data, which are under publication or already published. So, yeah, so understand that scenario, okay? So play the YouTube video. So this is the video. So we are talking both Ebola and NiPHA virus. Those are important. Ebola is not present in India. It's mostly African countries. Oh, I thought I am doing. So NiPHA virus is present in India. It's present in India, in Bangladesh, in Malaysia. Some outbreak happened in Singapore. So we are talking those important things. These two all are BSL-4 pathogens, very infectious. So please play the video. So the video will give some idea of what happened to the Ebola virus.
From the jungles of West Africa to the cities.
Dead bodies 15, 16, 17, 18.
The inside story of how the Ebola virus spread
It's pure craziness, like watching a zombie movie.
and the global failure to stop it.
The world has never faced anything like this. We've never prepared for it.
Ebola was not an exception. Ebola is a precedent.
I got a call saying that there's a worker on his way down to market. I hear and see the crowd of people screaming and shouting. I can see that he's afraid, so he has picked up a rock and is waving it around. He's followed by a lot of people. I guess you have to take him down. He's affecting our community, we don't know where he's going. But the pressure from the crowd is mounting, they're yelling at us. So the guys in the suits wrestle him to the ground and lift him into the back of the pickup. How did I end up here? Is this just a bad dream? But no, it wasn't. It was real.
In December 2013, the children of Meliandu village discovered hundreds of
Il dit, le village m'a tué, le village m'a tué, le village m'a tué, le village m'a tué. Il a commencé à crier.
Là, nous on se trouve à l'endroit où il m'a poussé. Soit funda ma pengima walero chaichina banaya banlandwa ha mavu china chuchuo fomi sina kolola
Khalil's blood would later be tested.
Je n'y avais pas pensé que c'était Ebola.
The government of Guinea had no idea how to respond. All previous Ebola outbreaks had occurred over 1,000 miles away. But the relief group Doctors Without Borders had decades.
We'll play a couple more minutes and then stop. Yeah. My point is like you can watch this whenever you're free and not feeling sleepy. It's a very like it in how break happens to body. If it happens in future like this, how
They already knew that like the existence of Ebola. So why did it take that long?
so Ebola outbreaks. So I my slides. So this is the
To be having outbreaks right before as well.
before they have, but there is not too much death. So same like in Kaisanur forest, right? Oh, a few died. You don't take care of it. So that's why, because it's transmitted through bat. Okay, now that what happened, it spreads so easily because everybody has a hemorrhage, bleeding, and everybody touching the blood. That's the thing say, like, the mother died, then somebody cleaning, they died, they went to the place to help, they contaminated other people. So that way it spread so many things. What? No, same. You can stop it, it's not working, right? Because so I'll tell you what it's the same virus though, but the reason is also the severity of the bat populations, right? We don't know when the earlier outbreak happens, how that happens. Maybe those bats are having a high loaded of viruses, and those people are consume too much virus, and the human population. So that is one thing. Second thing is maybe it is localized because the people died, nobody came and rescue, and maybe the localities, like, maybe it's an urban area and that's a rural area, right? So because the transmission does not happen so quickly, and that's why it stopped within some small group, even though, like, I think last year they have outbreak too, but there is some death, I'll tell you that. So that's maybe the reason also, like, it's confined to that because they have not transferred. It's all due to the transmission. So I came here, you touch this, and spread so quickly. That's happened. That Ebola virus happened so massively. But
why it's not spread to other countries or other
continents,
or other countries?
because it's very transmissible best. Those bats are mostly present there, Africa.
But it's a human to human also, it's a transmissible.
If through the blood, through the blood, not by. It should have a direct contact. If you have a direct contact with any mucus samples, saliva or blood, then it may happen. Not by sneezing, coughing, no. No, touching is not. But if you touch with the blood, then okay. So it'll work, right? Why it's working with you, not me? You played some trick there. So this you saw the day one already, right? So you saw the slide before day one. So inflammatory is very pathogenic, as well as so yeah, the NiPHA. So the virus, how it longs, biological Ebola virus, it's mononucleovirals. We already discussed mononucleovirals. You can see the paramyxoviridae, filoviridae, rhabdoviridae. So these are all together. So paramyxoviridae, we talk about those, you know, rabies, rhabdovirus, measles, canine distempers are here. These are not that important, but the filovirus is mostly the Ebola virus is here. And then respirovirus are here. The virus, it's an elongated virus. They have a glycoprotein, they have a matrix protein. And it's more specific for PhD, so I put all those. Those are there. So you can see the nucleic. So these are important. So Marburg, they come very from the more important. DRC for Dominican Republic, why they are also, see, um, if we have time, we'll talk on tomorrow. This is all. Here is the out. I told you it's only 12 confirms and so the question is why there is not big outbreak. It's mostly depend on the transmission, how the transmission happens, what the situation at that time. You know, that's happened. Like COVID also. Why the COVID has happened too far? Because the transmission happened so quickly. In SARS, because the COVID is also SARS, right? SARS happened several years. The question you can ask also, like, COVID-2 is so pathogenic, so easily transmissible, it is through pandemic. So same like, uh, Ebola virus. These cases, to and here is the picture of the the whole Africa and all those different symbols showing how many cases, what pieces happened, and those are listed here. Filovirus. This is the replications. So these are all more important is regions need endosomes bind with the lysosomes. Then again the same structural proteins inclusion that I'll show you did inclusion body. It happens on herpes virus also. There is inclusion body. So even that you can see under microscope the inclusion body. So virus makes a small kind of body where the virus resides, and that's called inclusion body. So they make an inclusion body where most of the genomic replication, nucleic acid assembly happening in that part. And then they become, total complete virus by the Golgi, and then exported outside. So this is a very, very nutshell what Ebola does. Ebola virus in this Indians innate comes very is the antiviral cytokines which comes very early for any viruses. Influenza. Innate immunity does not early. There is a lot of work done. Innate, if does not come early, adaptive immune response. Preventing does, I'll tell you tomorrow what is antigen presentation, because antigen need to be presented by the cells so that the T cell, CD4 cells can function, CD8 cell can do more cytokine effect and all. Function causes in case apoptosis. Here, like dendritic cells and CD4 cells help cell, they always interact. These are the core does not happen properly, antigen presentation does not happen. Virus actually blocks all those steps. You see the cross B cell development for T cell impairment, so that there is no clonal expansion. CD4 need to be clonal expansion for the better antigen presentation time. There is a T cell apoptosis happening. CD8s also can, it causes. CD4 is for B cell help we talked about yesterday. So there is no CD4 expansion, so B cell cannot get help also. And if B cell cannot get help, antibody. All immune functions as well as immunity, it causes dysfunction, both innate functions as well as adaptive functions. So this is the 3 Ps, okay? Talk about one children, right? The children get the bat and brought it to the village and then ate those bat, right? This is the index case. Those kind of people transferring from and to the village. So that is the index case, and that is the guy, the child. So he actually transmit all the everywhere, but it's in the village. Virus is always there. The bats is having the reservoir host. Bats is circulating the virus all the time. They don't die. I'll talk about why the bat does not die. And somebody asked. So bat is always harboring those viruses all the time. But also like other animals, chimpanzees, gorillas, and they are also intermediate hosts. They don't die, but they just taking the viruses. You saw the movie, like, the people kissing the gorillas, those things, but you're close to the gorillas, call those animals. Maybe possibilities that you get some point to the transmissions. So,
but it's not by contact, it's not.
Thing like if they injury in the heart and you just not carefully taking care of those animals and they say, okay, treatment on those things without wearing gloves or something, there's more possibilities. So because those animals harbor those viruses. So, so there is an outbreak happen. That's they discussed in the video. Transmit everywhere. But it's not like 100% lethality, right? So 30%, 40%. Some survive. But those carry the virus forever. Yeah, the virus is there forever. They have immune suppression or something. They go back again, they get treatment and then come back. So you have to be careful, those pets, because if kind of sexual intercourse or maybe some way you get the contamination with this person, there is possibility the virus.
So infection doesn't always need to be there.
Not necessarily. Able to cope up, yeah. It's not like, so general, like the virus try to hijack those 2 systems, right? Innate and adaptive. But for those, yeah, to generate this antibody, show you some monkey model study we have done. Or, military is an outbreak. Even like, ISON, NIH, I took the vaccine too for Ebola. I can take it or not take it, I just take it. So, for workers who are, because I'm the Ebola, so I can take the vaccine. I'm eligible to. It is it's like that's why like 60% or 60% they able to cope up, they able to somehow generate the antibody and they survive. So just So your question is, if the parents has, so yeah, so that means you are asking question like affected. You also get the, it's also like a infection happens. Of course, and maternal antibody.
Basically saying the immune system can be inherited.
Yeah. So that's normal. That's normal. So you see, that's a general idea. Like when, bless you. See, 6 feet. No, I'm just kidding. So when the mother is have a baby, right, and mother give the birth, all the maternal antibody transmitted through the cord blood to the baby. This is a general. So if the mother has some infection and they got antibody against the infection, go to the baby. Baby got protected those by the antibody. Sometimes what happens if the mother got infection during the time of pregnancy, either second trimester or third trimester, especially for herpes simplex. We worked a lot on herpes simplex. If the third trimester, the mother got an infection, there are more chance the baby can get herpes simplex also, the virus can transmit. HIV also the same. If it is happen in the later stage of pregnancy, the HIV also can go to the baby along with the antibody. So,
the immune system will not be inherited.
well, it means if they by then,
It will be inherited.
yeah, but no, even the mother if able to generate antibody enough, it will go there. That's a normal. Even when the mother is breastfeeding the baby, it's coming through the breast milk to the baby.
That's passive immunity.
Yeah. No, but it's still the mother's antibody is going, right? It's mother's
It's
antibody.
like the child will not be inherited from the mother. That means his immune system will not be trying to make antibodies.
Yes, it's going, the antibody going. The antibodies are going. Not the cells producing antibody going. Okay, make a difference. Memory cells present in the mother. Mother is having it. Mother is not sharing those cells, okay? Mother having, and the memory cells are mostly present in bone marrow and others. The cord blood is going, it's mostly the peripheral bloods. Whatever cells is there, neutrophil, eosinophil, basophil, macrophage, disease, they are going. But mostly the memory cells are present mostly in the bone marrow and other tissue. The tissue specific, they are not going to transfer. Antibody going, not the cell going. Is it clear? No. Antibody stay as long as the breastfeeding happening. So yeah, through the breastfeeding, it's going to the baby and it's protecting. Yeah. And until when the baby's immune system still naive, right? Yeah. So they are also maturing. And once they got some infection, then they get, and that's why they in the first 12 months and the next another year, like first 2 years I should say, that's why the baby get a lot of vaccine and boosters, so they can get vaccine against all the normal pathogens like measles and all those. They can generate them there because that's why the vaccine is. You are generating memory cells, so you should not get infection. Yeah, that helps. That helps. Yeah. If the mother survived from the Ebola, definitely there are more chance the baby can survive too, because the baby has the antibody come from mother. Virus, if it depends. Like if the viral level is very high, there is more chance it get transmitted. If the virus low or the undetectable, mostly the virus reside where? In the for mother, I'm not talking males. For mother, it may be in the cornea, in the brain, and some tissues. Yeah, it goes to the brain too. So in the tissues, in they have it. Okay, so the virus in the peripheral circulatory blood, it's not detectable. same like HIV. And in the eyes, because in the eye there is two different kinds. No blood flow there. So because there is no blood flow, I can stay here for longer. So that kind of organ, so many organs like blood, because blood can, the blood-brain barrier is there. So virus stay there happily. Uh, Viruses does not go transmit easily. For male, yes, it may transmit because it's transmitted through the semen. So the testis has the virus stay there all the time.
So the Ebola also enters the brain barrier?
Yeah, of course. That's why you go to brain. Whenever go to brain, no question, it go across it. Otherwise, I will go. Like, it needs a breakage to go in. So, right? So these are these are for early febrile phase, mild phase, and then you have a, and then mostly stages happen, there is hemorrhage. When the hemorrhage starts, it's very like 7 to 12 days. That's the phase where you can see that's critically ill, usually hypovolemic, often like confusion, seizures, those. And you see, as I said, like, so this arrows are, so this is the infection rate from 1%, one sick person, how many you can infect? That is called R0. So for example, hepatitis C, hepatitis A, those kind of like, it's only 2. RO is a 2, but you can say measles. One measles is through transmit 18 people. COVID is same like. One person, it can transmit 18 people. HIV, it's very low because it mostly needs sexual transmissions or intravenous drug users, those things. It's direct contact. Even kissing and all those, HIV does not transmit. It needs some mucus, some blood, cell-to-cell interaction, those things it needs to transmit. So Ebola is very less transmissible. That's another reason, like, some area it's focal, but that area question, like, definitely it's a different scenario, maybe the transmission, because everybody don't know. That's why the video says they don't know what is this, why people are dying, because then it was too late. Because the incubation period is 12 days. When the symptom will start, it's already late because the transmission already, that's why the video says it's already crossed more than 1,000 miles, the virus already, before the symptom starts. Experiment and some monkey model to understand the initial phase, like so these are groups where we did monkeys, the virus, and then he collected different samples. Different groups we have here. Some we are collecting after the infection 3 days, some are 4 days, some are 6 days. So that you can see the study day 6, study day 4, study day 5, and some we are not doing collecting samples, we are collecting at the end stage. And want to see what is happening. There's the red blood cell phagocytosis. What happens? And that's we have seen. There's a lymph node also. We can see the neutrophils and red blood cells and all the presence here. These are the inclusion bodies are present. I mentioned that the cells, the Ebola virus makes inclusion body. Herpes another virus which make inclusion body. That's the characteristics. They cause hemophagocytosis. The once it got infected, so the tissue, yeah. So the RBC may be tagged with the antigen or the Ebola is there, and the macrophage engulf the whole red blood cells. And it is very common for Ebola virus. You can see the big cells, and inside the red blood cells is there. It is normally also happen. The phagocyte, like, a red blood cells is very going to die or something. The macrophage function is like this also, which is, you probably heard who got the Nobel Prize for autophagy. Autophagy, people, I think some people know that. Autophagy is like fasting, right? We do, some people do also, I sometimes try to, 14 to 16 hours fasting. Like I took like 7 o'clock until noon, I'm not ate anything. Body has a mechanism, the macrophages especially, any dead cells or any cells going to die, they engulf it and clear it. So this is kind of body self-cleaning, like a microwave self-cleaning, anything. Body has a self-cleaning mechanism. So macrophage take those bad cells and clean it up if you do fasting. If you don't do fasting, but the cells are always active, then macrophage cannot do those things. But by fasting, that's called autophagy, and people got, I think the Japanese guy who got the Nobel for this, and it is truly fact. Same all the time, macrophages do the same thing. A red blood cell is going to die or something, it will engulf so that they can eliminate those bad cells. It is common also, but we saw that in Ebola it is more prevalent, and that means it is a hemorrhagic disease. So red blood cells are dying also. So that's why the macrophages try to clean up all bad red blood cells, and that's why it's doing phagocytosis. Here another example. So we looked towards soluble CD163. It is a marker of macrophage activations. Macrophage CDC163 is a marker of macrophage, but also you can measure the soluble part of it. It's a ligand which also expressed in the blood, okay, plasma, and you can measure it by ELISA. And you can see there's upregulations. As the Ebola virus got increased, there is upregulation here. It is the flow cytometry data, showing the CD163, which is the cells expressing the macrophage. It increased of the macrophage populations. And there is also the macrophage is shading those CD163 marker in the serum, semen. Yeah. So yeah, we put JID. What does it stand for, CD163? It's a macrophage marker. So there are a lot of CDs. C means cluster differentiation. So it's a cluster differentiation, it's a marker. Yeah, so look it over when you have time. So based on the cluster differentiation, okay, good point. I have not discussed that. So say cluster differentiation is a marker. I should say it's a biomarker to define what cell it is. Like the same thing, like we talked about CD4. It's a CD4, CD, cluster differentiation 4. Okay? CD8, it's the same, cluster differentiation 8. CD163 is cluster differentiation 163. CD8 only expressed in the T cells. So when we run like flow or anything, yeah, we say, okay, it's a T cell, CD4 T cell.
What is the role of these proteins?
Hmm?
This is a protein, right?
Of course, yeah.
What is the role of it?
Surface expression. So not necessarily everybody will express on the surface, right? So like 163, they express on the surface. If you see, like, we talk about another terminology, proliferating, cell proliferating. I said cell are growing. The marker is Ki67, okay? But they don't express surface. They are intracellular. So you have to do different kind of staining to identify those. Then cell express, okay, we talk about like CD8 expressing granzyme B. We talked about like a little bit, if you remember, CD107, those things. Killing activity, right? We talked about CD8 has a killing activity. How you know killing? Do they have a gun or something? No, they have a like CD107 expression. CD107 is called degranulation molecule. Granzyme B, that also has a killing function, granzyme. So it's an enzyme that kills the cells. So how you know those? Those are not expressed in the surface, they are intracellular. We have to do some intracellular staining to find out those markers.
Those proteins induce apoptosis in cells?
No, the apoptosis is separate.
Okay.
They directly kill. They release, so they bind, like CD8, okay, this is infected cells, this is CD8 cells, it binds, and this is bad cell, it releases. And then that granzyme B is the protein, it digests whatever inside is there and kills. That's the way it is. And also CD107 degranulation molecule means it increases the pore. If the cell has cell wall, has a pore, right? It increases the porosity. If the cell increases porosity means what happens? The cell inside the Golgi may come out, or the pH has changed, the cell become bad and cell die. So they do that kind of functions. Okay. So overall, CD has a different. It is mostly used to define. It's called biomarker. It mostly used to define cells. So 107, you can say what is CD11C? It's a DC marker. Okay, dendritic cells. Okay, so there are two markers mostly used, like CD111 and CD123. One is a monocyte-derived disease, another is a myeloid-derived disease. Okay, this is a myeloid-derived disease. Okay, myeloid, it's a myeloid cell. We can talk later outside. So myeloid-derived disease. DC is also derived from different cells, monocyte or myeloid cells. Yeah. So you can ask definitely ready. So basically, that you saw increase in macrophage. Okay, the overall point. So then again here we put another 56. All those are CDs. Okay, so CD56 mostly is NK marker, natural killer cells. We talked about natural killer, but this is another marker which you use for marker to see whether the cells are activating or not. Okay, there is activated, or it's not sleeping, it's activated. Like this way, in the night we sleep, and the morning we got activated. Maybe somebody take a cup of tea, or somebody coffee, or a cigarette, right? So they got activated. So then HLA-DR is that kind of marker. The cell got activated. So but HLA-DR is a late activation marker. If you see early activation, you use CD69. That's another CD marker. So we differentiate the bed. Here you can see HLA-DR expression going down. If you remember the couple before, like we saw that in Ebola virus, NK cells cause apoptosis and all this. Exactly, you can see here. Double positive, right? They're positive for HLA-DR, positive for 56. So they, activated NK cells. But we have a lot of cells here. Those cells are. So definitely Ebola virus, when it's happening, it starts early. After 2 days, it's going down very early. Why we did the monkey study? Because you cannot do this in human, right? Yeah, we don't know when they eat the bat and when the transmission happens. So all those things. That's why the animal model is important, to pinpoint where the change is happening.
But NK cells are important though for innate immunity.
Of course.
But they main components to have such a kind of like a dramatic severe disease.
But NK is very important initially, right? It's a natural killer. It will kill all those infected cells. Indiscriminate, yes. Now they have a study. There is a group, and we have also seen there is an antigen-specific NK cells also that develop a little later. Initially, what comes is irrespective of what antigen you have. If it is foreign, they say bodies find out, why this person came to me. I have to get rid of it. NK comes in. The antigen specific, if they, after infection or something, then there is, you generate the antigen specific NK. Then if you get again the virus, then those NK functions as well as some non-specific also functions.
But I'm puzzled, like in case of HIV, it directly targets one of the major component of immune system, like the CD4. But it takes sort of like a time period to kill the infected person. But here, although it's important in case it's getting reduced, but the fatality is very fast, kind of like maybe within weeks, maybe at the max.
That's a lentivirus. It's a slow, right? The HIV is very slow. Yeah, even we studied a lot of NK. We don't see any changes in HIV. There is none. NK remains totally stable. NK that does not touch at all. Only if you see, I have presented my data. I think last time when you presented the CD4, you can see the similar kind because HIV targets CD4. You can see drastically by 9 days after infection, it goes to baseline. Right, like from for example, like 400 cells per microliter of blood, it comes to 50, and within 10 days.
So no studies have been done with respect to all these T cells or any other things.
What? Ebola?
In Ebola.
Yeah, we did. I don't know. Yeah, we did B. We looked over in case of. You can say CD4 has changed, but
Major part of the immune system is intact despite the infection.
CD4 has no change. CD8 has no change. There is change in the total lymphocyte populations. There is change in the monocyte platelets populations because they hemorrhage. There is platelets go down. There is change in the fibrin. I'll show you. This is another. Just to show, I'll come that in a second. Okay.
Maybe I can interpret that like as long as it doesn't go to the brain or doesn't cause hemorrhage, otherwise it's not severe disease.
Which one? Ebola? Ebola is that's the initial important. Like, that if you survive, if you're able to cross that 10, 12 days, you're fine. But the initial is very drastic because you have a hemorrhage. If you don't get enough, I'll talk about it. Like if you don't get a fluid therapy, if you don't get a blood transfusion, then you are out.
So it's dangerous because it goes to the brain.
Yeah, it's hemorrhage. It's.
Yeah, but going to the brain is most kind of like severe effect. That one.
Brain, HIV also goes. HIV also goes. West Nile goes. That is not that severe. The severity comes. It's a shock. I have a hemorrhage happening. The blood. I'm losing blood. Same like diarrhea. I have to drink water, right? So same like I'm losing blood. I need some either fluid therapy or electrolyte therapy because my electrolyte balance is getting bad. My blood volume is getting thicker because I don't have enough blood. And that's why I need blood, I need electrolytes, I need those things. If that's why that is the supportive therapy, I'm coming there. If you support those way, you may survive. If you don't support this, despite it goes to the brain or not, you may die because body wants to maintain that, that same structure what was before. Same like blood volume, same cells, same thing, blood transfusion, you give blood. That's mostly the supportive therapy is. HIV also goes to the brain, West Nile goes to the brain. So many, Kaisenol forest also goes to the brain. But brain is okay, people got encephalitis, that's a different treatment you can do. But that's the major hemorrhagic disease. This is the slide just to show, we find the virus everywhere. Those are the positive cells. So is this the P40? We mentioned those are the proteins present, and you can see those. There is a hemorrhage also. We have shown here the top left, top right, and the bottom also showing that the virus is present. And you can see the testicular hemorrhage present. So here another slide just to showing how the Ebola virus detected in megakaryocytes, but not in erythroid and myeloid cells. We use some marker to differentiate whether it is erythroid and myeloid, and we don't see it, but we can see another showing that inclusion is bought in those.
This data is derived from the animal model.
Yeah, this is from bone marrow, and we published in the American Journal of Pathology. Yeah, so this bone marrow, we have seen the virus is present there, and it's infecting megakaryocytes and not the erythroid myeloid hematopoietic cells. What? So normally, hematopoietic cells gives rise to all those cells, right? So we want to see whether it is infecting the stem cells or they are progenitor cells. So that's the idea is. So it does not infect the stem cells, yeah. And you can show that we did transmission electron microscopy. You can see it's present in the bone marrow. This is the virus, just to show.
Could it be that that stemness property is what's protecting those cells?
Hmm?
Could it be that the stem cells, the stemness of those cells is what's protecting them from those viruses? Could it be like that?
No, I present in different way. I said it's not affecting the stem cells. Stem cell, so hematopoietic stem cell gave rise to megakaryocyte, other karyocytes, right? So the stem cell does not get infected, but when they matured, they affect the mature cells, not the progenitor cell. Yeah.
So my question is, the hematopoietic
Stemness remains same.
will have some amount of certain genes that cause that.
Definitely. Oh, of course.
Could it be protecting?
Right. Yeah.
Okay.
That's true. Yeah. So this slide is very easy to show that every tissue measures. We measure every tissue. The virus is present everywhere, whether it is ovary and cerebellum. So the brain, brain's present. Lymphatic different organs present. Kidney, bladder, thymus, axillary lymph node. Name it, they have it. Those kind of things. Oh. What? Cerebellum is low, yeah. I mean, so there is cerebellum with whatever which select, right? There are a lot of animals, right? So majority are there. So see, there is we are undetectable, right? But spleen. My point is, because this is like a heterogeneous group, right? Monkeys. It's not like mice all are homogeneous. Same phylogenetic, same genetic makeup. So monkeys are different genetic makeup. Same like humans. So this data represents like a human, because also human, I may have, if I have a HIV, maybe all those negative, but maybe this positive. That's my point is, like, that's the percentage, like 15%. My point is, see the virus all the tissue, despite 100% in all, but you should see somewhere, right?
So it goes to everywhere.
Exactly, that's the point. It goes everywhere, it stays there. So I'm not going, so we look at thing. So we said that the virus like a, so it causes triglycerides increase, and then loss of activity cell is caused to see. It causes those things, cytokine storm, causes macrophage activations, causes T cell activations. We made, but there is no change in the CD4 functions. Pancytopenia, organomegaly, fever, and all those. Actually, macrophage, I'm not. It's little harder maybe, but this is the mechanism for showing how the macrophage activation happens. My idea is to say that this is the mechanism how macrophage activation happened, and we saw the same mechanism we saw in the Ebola. So that's why Ebola causes macrophage activation. It's a macrophage activation syndrome, and all the way what we are saying, like CD163 is in, RBC is involved, ferritin is involved, and all those anti-inflammatory cytokine upregulations. We have seen everything the same pathway. So this is the risk: mortality, high morbidity, mortality. Mortality. After infection, also, if you survive, this is everywhere. It's in the semen. It's also in the uterus. What we mentioned. It's in the brain. So the virus is everywhere. Even it's milk virus may transmit too. So it is present there. So the diagnosis, kind of symptoms we saw in malaria, those are prevalent in malaria typhoid. So it's assays, detection assays, and virus isolations. These are all those things it does. If I mentioned, the virus won't affect all these organs, so that means all organ abnormality you can see, starting from the brain, starting from eye, all those. So what mostly it's a symptomatic treatment. So if you have depression, you take psychological. If you have a headache, take some analgesic. That kind of thing. If you have a lot of water and those are diarrhea, dehydrate, those are. But there are two monoclonal antibodies are very is already established Ebola infected patients because those have survived. So they isolated those antibody, then it's been cloned and grown multiple times, and they made it like a stock, and those can be used for only for the neonates of confirmed Ebola virus patient or the patients with are PCR positive. So for them only, you can use those monoclonal antibodies to treat them.
And they are effective.
Yeah, they're very effective. So same like, heard about the COVID-19, right? The monoclonal antibody treatment, and that's another. Like, if a severe case, just take those antibody that, antibody bind with the antigens, and then it's get phagocytosed and get rid of from the body. So again, from the question, how to keep the body function weights, and management of complication, those things. So vaccines not, it has and behavior. We also can go international coordination group and the vaccine trial, other groundbreaking vaccine trial. Have, like frontiers people, Indians. The Ebola does not infect Indians or others, right? So we don't need it. Well, so policy and I part. So their Ministry of Health, they can, I don't know those. This is just to show, like, their recent outbreak, what happened 4th September, some outbreak already happened. There's some death already, like 14 suspected cases and death. I think the death, 14 died out of 27 or 20, yeah. So already, so this is happening all the time in regions. But the only things is, the PPE, you can see CDC, WHO website helps when you handle that kind of patients. Just for your info, you have to cover your body. So this is the final. There is no exposed part in your body that can touch anything or infectious. Okay, shall I go this? Huh? Nipah virus, will talk about Nipah virus. So, and Nipah virus is very prevalent in India.
I'll be taking a little bit of study, but maybe, yeah. So for lunch, I think, how long
Yeah.
this will go?
It will take a while. more.
Soft lunch? Okay. We say
Well, you can finish tomorrow.
yeah.
Yeah, depending on time. If yeah.
We can break for lunch now.
Okay. Say me again. Sorry, the sound is disturbing. We don't know, right? So the outbreak happened does not mean that virus started right there. So because it may happen so many years ago, right? Nobody knows, and it's still under bat, replicating over and over and over. And suddenly it goes to human, expose, and then we find out, right? Yeah. So right.
And suddenly it is there. And the vaccines or the measures which was working previously is of no use today. Is it because it is keep on changing with respect to its capsuleers or the spike protein mutations? Or
Of course, yeah. You're talking corona, right? Is COVID, right? Yeah, same thing. Because virus mutates and changes, right? And the vaccine is working before. That's why you need, you get say, oh, you have to take another boost or another different kind of vaccine, little changes, just to keep you up to that, like you are covered. Because the spike protein, especially corona, it changes so much. It's so much variants are there. Some are very pathogenic. Some becomes mild, which you don't care actually. But yeah, if it is pathogenic, they find it's very, you need to take another vaccinations, maybe boosting.
and simultaneously the immune system of us is depleting with respect to them.
No, it's not depleting. So it's how I'll say, your body has a cells, right? Like for example, you take 100 cells, hypothetical, right? I have a body 100 B. So now I took a vaccine today with COVID. From 100, there are 100 are some are naive B cells, some are memory B cells. So from 100, maybe 10, I got memory B cells, which got plasma cells and producing antibody. Okay, I have that throughout my life. Okay, I took the vaccine, it's there. Then the health says, oh, there is a variant and we made a new vaccine because it's different from that. Okay, there is a different spike. A lot of mutation have changes. Okay, I took that vaccine, so I have 90 cells still there, right? Naive cells, maybe 50 naive cells still there, right? Out of 100. 10 already made vaccine when producing antibody. But I still have a lot of naive cells. Now I got that vaccine so that some naive cells, again, memory cells plus 10 memory cells, got that. So I have now one from the previous one and the 10 for the new. That all the 10 there, it is there. It's already there. And the 10 got for the new is now there forever until you take the new variant comes or something. So that's why you generate memory cells in your life and it's there. But the more mutation happens, yeah, something new, and then you have to take the vaccine to generate another memory populations. But it does not, by generating another new subset of populations, you are not diminishing the previous one. It's there already. Maybe they are inactive, they are not doing, oh, there is nothing to do, I'm just sitting there. But it's okay. Because they are memory, they all stayed in that same way. Because they'll not be active until they see that same antigen again. They'll remain dormant. That's why they say plasma cells live longer and they dormant. Where they dormant, they mostly remain in the bone marrow. But when with the blood, I got the same old infection, maybe 10 years later, and by blood it goes through and go to the bone marrow, then bone marrow B cells saw, oh, there is a new thing came. The same 10 years ago that came again. Then those memories try to proliferate and makes more antibodies. Yeah, so there is no decrease or increase, rather than you generating more memory populations and fighting for all kind of strains. Yeah.
Can Ebola be called
it does not cause immunodeficiency virus because it does not deplete completely. We saw the NK cell deplete, right? But the NK is not a major cells for the T and B cells. Yeah, because it's fall under the innate cells. So when it is adaptive, when you
Our
it
body
is
has
compromised, so it's immune. Yeah, I said like all vaccines so far today, from your childhood and everything, who gave protections? The B cells, not NK. Who give protection? The T cells. Those are the two major. And when we deplete any of them, when the virus affect any of them, then we can say it's a immunocompromised or immunodeficient virus. But NK, no. Type 1 interferon, no. I don't care about those are innate. Maybe they will come back at a certain point. But initially they are down, maybe they will come back because they are not directly targeting those cells. But when HIV, they are directly targeting CD4. Even though like 10 years after virus suppression, drug suppression, so mostly they are taking drugs, right, to suppress the virus. But if suppose you are not interested, you want to check yourself, okay, I'm not taking drugs, let's see what happens. The virus will come again and kill your CD4 again. So that means it is always.
Why not use antivirals for Ebola?
There is none.
Can't repurpose the ones that are there?
There's no repurpose has not tried but does not work. Yeah, there is literature there, so many there, it does not work. So that's why the monoclonal antibodies and they have vaccines and supportive therapy, that's it. Good question. For that, you have to take the same strategy like smallpox, right? Because and also foot and mouth disease virus in cattle. They have done this by some country like Europe, for example. Let's talk about smallpox, right? How they did eradication, vaccination.
No, she's talking about an infected person who has
What year?
like the infection will remain in the person, right?
Right.
She's asking, is there a way to remove it completely?
No. Because that's I think we mentioned for HIV too. It is very difficult to destroy any resident viruses hiding some tissues. Because the drug cannot go there in the tissue to kill those viruses. So that's why, despite HIV is the treatment is much better now. Like every 6 months you are taking one medicine, but still virus is sitting maybe some lymph node, maybe somewhere, maybe in the brain. It is there. You cannot completely eradicate. Same like Ebola, the virus is there somewhere, but you cannot cure them. You only have, because you have a good antibody in your body, because you cured from cell, you naturally developed your antibody, naturally gave very good T cells, and you live that way. There is no way to cure completely the virus, because the places they are residing, like eye fluid, the drug cannot go there. Brain, some of the part of brain, so that the drug cannot reach there. They cannot. That some drugs cannot cross the blood-brain barrier until you put the injection on the CSF, cerebrospinal fluid, and drug directly. Then it can. But which person wants that kind of treatment, right? People always prefer to get oral drugs and all those things, right? Even for like a cancer patient, like taking the radiotherapy and others, it's a very risky things, and it has a lot of side effect and a lot of things. Even though like how specialized, you do the right CSF inoculations, and you want to not generating another blood, disturbing the blood-brain barrier, right? So those are the questions are there. To do that kind of special treatments and cure it. Okay, thank you.